Okay, so you guys remember exactly what causes the pH to go down in the blood during acidosis, and that's because two of these ketone bodies have acidic groups on them, and that's what causes the pH to go down. Okay, so I think now is a good time to kind of review some of the figures in your book. And we'll start off very basic, and then we'll sort of build up here. So this is something that I had talked about before when we were talking about glucagon a long time ago. We were talking about glucagon and how glucagon is secreted during times of starvation, and that basically mobilizes the liver to produce glucose, and the glucose then is going to be dumped into the blood to feed other organs, especially the brain. Now, we just talked about when you have high glucose, so you've just had a nice meal, and so now you have high blood glucose. What that does is that causes secretion of insulin, and insulin then, at least for the liver, will cause the liver to say, oh, we have times of plenty, okay, so let's go ahead and store the glucose in the form of glycogen, okay, and that's what the liver does. So it will be able to use this glycogen to feed the other organs in times when there's not very much glucose around. Of course, the insulin affects other organs as well, okay, and the insulin will affect, for example, muscles, and it will cause those muscles to take in the glucose and store that as glycogen, and also to use some of that for energy purposes. Okay. Let me just, there's another figure here. Yes, this is what I wanted to go over. So this is on page 769 of your book, right here. Okay. It's kind of nice because it summarizes what I've been talking about over the last few days. So let's start off here with the high blood glucose. And so what's happening in the liver is, as I said before, the glucose is going to get converted to glycogen. And then it says here it's going to get converted to fatty acids. And that's kind of that is interesting. So what happens is that glucose can go through glycolysis, and then eventually goes through pyruvate dehydrogenase complex. And the product of pyruvate dehydrogenase complex, what's the carbon product of pyruvate dehydrogenase complex? I didn't remember. Acetyl-CoA, very good. And acetyl-CoA is the building block for fatty acid synthesis. Okay, so you can actually make fatty acids. Now, we didn't talk in detail about that, but you should know that in general, acetyl-CoA is the building blocks for fatty acids, and those fatty acids can be converted into triacylglycerols, and those triacylglycerols then can be secreted as very low density lipoproteins to the body. Okay, so that's what happens when you have a meal. Adipose tissue, we've talked about this quite a bit, how the fatty acids are delivered to the adipose tissue in the form of one type of lipoprotein. Does anyone remember the type of lipoprotein that delivers fatty acids to the adipocyte? Actually, it delivers it in the form of triacylglycerols. Anyone remember? Chylomicrons, very good. So chylomicrons will deliver this to the adipose tissue. Actually, there is there's a brief hydrolysis of the triacylglycerols from the chylomicrons where they become fatty acids, and they're reassembled inside the adipose tissue as triacylglycerols, also known as TAGs. After having a nice meal, the skeletal muscle will take in the glucose, and it will use it for glycogen storage, okay? A little bit of it will be used for glycolysis, and that glycolysis ends up in pyruvate, okay? That's the end product of glycolysis, but then one more step it can take, and that is to go from pyruvate to lactate, and I talked about the importance of that reaction. What's the importance of the reaction of going from pyruvate to lactate? That can go back to the Cori cycle. Very good. But immediately, what does this provide when you go from pyruvate to lactate inside the skeletal muscle? NAD plus, right? NAD plus, right? And that NAD plus will be used, recycled back into glycolysis. Exactly. That's really the reason. And of course, yeast do something different, right? Okay, very good, very good. And then the fatty acids are, after a meal, the fatty acids are taken into the heart. Sorry, just move this down here. Taken to the heart, and we're going to go through beta oxidation. What is the carbon product of beta oxidation? Remember, the carbon product of beta oxidation. I know what it says here, but you and I know, what is the carbon product of beta oxidation? The final product of beta oxidation. Where? The carbon product of beta oxidation. Fatty acid is the substrate. Acetyl-CoA. Acetyl-CoA is the final product of beta oxidation. The final carbon product of beta oxidation. But why is it saying CO2 and H2O? What are they talking about here? What's going on here? Why do they say this? CO2 and H2O. Where do you guys think that acetyl-CoA goes to? Citric acid cycle. So what they really should have said here is beta oxidation plus citric acid cycle, right? And then this would be accurate. Okay, very good, very good. And then of course the brain cannot accept fatty acids. I've said that several times, okay? It can take in glucose, and it goes through glycolysis, citric acid cycle, this is correct, and then it's gonna make CO2 and water. Okay. By the way, when they say water here, where's that water coming from? We know where the CO2 is coming from. That's coming from acetyl-CoA. Where's that water coming from? Beautiful answer. Beautiful answer. The electron transport chain. Yes, the electron transport chain. And what is the substrate for that water? O2. Very good. O2 and some protons that are hanging around and some electrons. Very good. Very good. So we know where that water is coming from. We know where that water is coming from. Okay. Next, we have low blood glucose. Okay, so this is between meals. You know, you're sleeping at night. Okay, you got to bed at 10 o'clock, and at 4 o'clock in the morning, you know, this is the kind of thing that's going to be happening, right? Okay, we got the fatty acids. They're going to be now the liver kicks into action here, right? So this is when the liver starts to say, okay, I need to help out the other organs. So fatty acids undergo beta oxidation as well as citric acid cycle in order to produce CO2 and H2O, of course, and it's providing energy for this. Okay, glycogen is going to be broken down into glucose through glycogenolysis, and that glucose is going to be exported to the rest of the organs out there. I should also say, and it's not listed here, but there's gluconeogenesis happening as well. Gluconeogenesis is happening in order to produce glucose, and that glucose will also be exported as well. So there's a number of things happening here in the liver between meals. Triacylglycerols are being broken down in the adipose tissue and delivered by serum albumin. We have a lot of serum albumin in our blood that carries the fatty acids to other organs, okay? And also, I guess, glycerol is also being made. But the fatty acids primarily being taken from our adipose tissue and being delivered to our muscles and to our heart in order for them to use, those organs to use fatty acids. Skeletal muscle, it's a consumer, okay? So it's going to consume these fatty acids, undergo beta oxidation. Oh, I didn't know that. Okay, so I guess you're going to produce a little bit of ketone bodies. I'm not sure if that's exactly right, but for sure beta oxidation. And then we've got the heart undergoes beta oxidation and citric acid cycle. And the brain. The brain is a consumer. It's going to take glucose and make CO2 and H2O. Okay. Then let's go under the conditions where you haven't eaten for a long time. Okay, a long time. Let's just start off here with the fatty acids. They're going to be converted into what was the 2-carbon product after beta oxidation? People? Acetyl-CoA, great. And then acetyl-CoA goes through ketogenesis to make what are the three products that make up ketone bodies? One of them is very easy. Acetone, that's the easiest one, okay? And then acetyl-acetate, very good. Beta-hydroxy-gluteryl-CoA, okay, something like that. Okay, so there's 3 of them there. Okay, and those ketone bodies are important because those get sent to different parts of the body as an alternative energy source. Adipose tissue, it's still going to be depleted here. Okay, so we got these fat cells are going to start to shrink. Okay, the skeletal muscle. Let's just look at fatty acids. I guess they can also do some kind of ketone body formation. I didn't know that, but anyway, I guess skeletal muscles can also do ketone body formation. And heart is a consumer, so the heart will take in the ketone bodies and make them into what? What does the heart do to those ketone bodies in order to get them to go into the citric acid cycle? Acetyl-CoA, very good. So it breaks down these ketone bodies or converts them into acetyl-CoA for the citric acid cycle, and the same thing with the brain. The brain will do the same thing. And then we also have amino acids that are broken down. I gave you one example of where alanine can be converted into lactate, and the lactate actually can be converted into glucose through gluconeogenesis. I just gave you just one example with alanine, but I think there are other amino acids that can do this as well. And then the skeletal muscle can take proteins, hydrolyze them, and make amino acids. These amino acids then can get sent to the liver in order to make more glucose. Okay, so we didn't talk very much about proteins, but proteins can also be an alternative energy source during times of starvation. Okay, I'm going to let me just go over just a couple more diagrams here. This one you have seen before. This is what's happening in the liver when you have glucagon. So glucagon, remember, happens when you are I guess between meals. Okay, you make this cyclic AMP. So glucagon will bind to the surface of the liver. You will make cyclic AMP inside the liver cell. And I just want to go over this one pathway, not all of them, just this one pathway. And then you have protein kinase A, which gets activated, and it will convert phosphofructokinase 2 to fructose 2,6-bisphosphatase. And what happens then is that this regulatory molecule, fructose 2,6-bisphosphate, goes down, and that will decrease phosphofructokinase, which is the glycolysis pathway. It will increase the activity of the gluconeogenesis pathway. And so what happens here is that you get a decrease in glycolysis and an increase in gluconeogenesis, and you need that in the liver in order to make glucose to export to the cell. Just this one pathway is all I'm interested in. Okay. I want to contrast that to another pathway, okay, and that's a pathway found here, and this occurs in lots of in cases when you have insulin. So when you have insulin, okay, you're actually going to activate this enzyme, which is on page 762 of your book, okay? And this enzyme, what it does is it's AMP-dependent kinase. This is also activated by high levels of AMP compared to ATP. So when the ratio of AMP to ATP goes high, in other words, low energy charge, all of these will activate AMP-dependent kinase. I'm only interested in just two pathways here. One is this pathway where you get phosphorylation of, again, this enzyme, phosphofructokinase 2, fructose bisphosphatase 2. That's that regulatory enzyme. But this phosphorylation occurs at a different site, which your book doesn't make clear. It's a different site than the pKa site. And in this case, what happens is that you get a increase in fructose 2,6 bisphosphate, and that's going to increase glycolysis and decrease gluconeogenesis. Okay, so this is what's happening here. The other thing that happens is because you've just had a meal. Okay, so what else is going to happen is that you're going to take a receptor. This is actually a transporter called GLUT4, and this transporter gets mobilized to the cell surface. So now the cell can take in glucose. Okay, so only this I want you to understand, and this one down here. I mean, all these other things are important, but it gets too complicated. Okay, you just had a meal, insulin secreted, AMP-dependent kinase gets activated. You're going to increase glycolysis, decrease gluconeogenesis, okay? And you're also going to, if this is a muscle cell, you're going to move the glucose transporter from internal stores to the surface so that now the muscle can take in glucose. And this is also triggered by low energy charge as well. Okay. We're going to stop here. I had other stuff, but I think we're gonna stop here in terms of material. So I know that we were supposed to get up to 18, and we did. There may be some problems in the back of the book that I assigned that aren't related to what I talked about during lecture. You can ignore those problems, okay? But the ones that concentrate on what I talked about in lecture, do not ignore those problems. I think we will now have a review of the syllabus. So we'll go ahead and do that. Let's see, where is that syllabus? Here we go. There we go. Oh, that's cool. Okay. So, first of all, I think this is correct. We have a final exam on Wednesday, June 8th, from 10:45 to 1:15. It will be held here. Okay.
So if the finals out of 200 points, will it be like 100 new material, 50 first exam, 50 second exam?
Yeah, at least 50, since we haven't had a midterm in a while, it's been a while, at least 50% of the material will be on new material since the midterm, at least, okay? The other 50%, or maybe 40%, will be on material before the last midterm. Okay, we had talked about enzyme rates. We talked about first order and second order reactions. There were a couple of sapling learning problems. Oh, that reminds me, sapling learning. There was a request to allow the sapling learning problems to be open for practice. And I've done that. So if you guys go to sapling learning today, you will see that all the problems are available for you to practice. Okay. So we talked about different order reactions. We talked about the reaction coordinate diagram as a way to sort of see how thermodynamics as well as kinetics work. Thermodynamics in terms of the difference between the initial delta G, the initial energy level versus the final energy level. That's thermodynamics. The kinetics, of course, is dictated by the height of the activation energy, right? And of course, you guys know that enzymes have been evolutionarily honed to be able to lower that energy of activation, which is great, okay? I also mentioned that there are some of these reaction coordinate diagrams that are a little bit more tricky than others. They have like a little dip, and then so they go up, and there's a little dip, then it goes back down. Okay, that little dip then is going to be something called an intermediate state, and it's often marked by a covalent bond between the substrate or substrate intermediate and the enzyme. Okay, at the very top of the energy of activation, we call that the transition state. We talked about Michaelis-Menten kinetics. We did talk about the induced fit model versus the conformational selection model. So we talked about that. We talked about Michaelis-Menten kinetics, how to draw a Michaelis-Menten curve, which is going to be initial velocity versus substrate concentration. We also talked about how to convert that curve into a straight line using the Lineweaver-Burke plot, also known as the double reciprocal plot. And those plots are very, very useful for distinguishing between different types of inhibition. Don't forget to bring a ruler for the final exam. That reminds me, okay? So don't forget to do that, just in case, okay? All right. We talked about serine proteases quite a bit. I went over in detail the mechanism of, I think, chymotrypsin, okay? We talked about the specificity pocket versus the catalytic site. We also talked about the allosteric site, which is another site completely that can control the enzyme. And the allosteric site can be controlled by inhibitors or activators. It can also be controlled by post-translational modifications, and the classic example is phosphorylation. We talked about K-cat, which is turnover number. We talked about K-cat over Km, which is enzyme efficiency. And here again is Lineweaver-Burke plot. So we did talk about those types. Oh, one thing also we talked about was irreversible inhibition. And that's typically done by a covalent bond formed between the inhibitor and the enzyme. And in that case, what happens is that you have to basically degrade the enzyme completely and then rebuild it from amino acids. Okay? And that rebuilding process, you'll go over that in Chem 431C, or if it's equivalent next quarter. Enzyme classifications. We went over enzyme classifications in detail, and then I kept on talking about that throughout the course to kind of get you to be thinking about what those really mean. The hydrolases, the ligases, the lyases, the oxidoreductases, right? The transferases. So you guys sort of have that sort of embedded in your mind. When you look at reaction now, you'll be able to distinguish those from each other. We also talked about how oxidation number is a good way to determine whether you have an oxidoreductase or not. So figuring out oxidation number. And there were a couple of problems, at least one problem on that, and so that's very important. Okay. We talked about how there's product inhibition. So the immediate product of the enzyme reaction can, if it builds up to too much of a high level, it can inhibit the reaction from going forward. This is Le Chatelier's principle, by the way. Okay, and also there's something called feedback control, where you've had several steps, A to B to C to D to E to F. In other words, several enzyme steps. Okay, and the final product, then can come back and inhibit one of the initial steps in the reaction. So we call that feedback control. Allostery is kind of an old term, but basically what it means is conformational control of the active site by something binding to the allosteric site. And a classic example is when, there's a number of them, but anyway, when CO2 binds to hemoglobin, it changes the ability of the hemoglobin to bind to oxygen. Okay. All right, then we had a quiz. Okay, then after that, we talked about covalent modifications. And I just listed some. I don't think I actually talked about these too much. I think phosphorylation is probably the biggest one you need to know. You should understand that mechanism, how ATP can donate a phosphate to a number of different substrates, including serines on proteins. Okay. And I mentioned methylation and acetylation, but I didn't go into those. So you don't have to know those. I talked about metabolism in general and how that can be broken up into two different classes, catabolism and anabolism, and how catabolism is energy consuming, net. Okay, so not every step needs to be energy consuming, but at the end of the day, I'm sorry, let me take that back. Catabolism is energy producing. Sorry about that. Energy producing. In other words, we're making ATP. And in glycolysis, it's a little bit complicated. You have an investment phase where you have to use a couple of ATP molecules, but net, you get 2 molecules of ATP for every glucose that you put into the system. Anabolism, on the other hand, tends to be, or it uses energy, so there'll be a net energy consumption, and it is reductive. In other words, we use NADPH in order to reduce things for anabolism. And so you should understand the energy curve, the energy charge curves that I showed you early on in the quarter. I did talk about energy yield for sure, in terms of energy yield from beta oxidation, from glycolysis, from citric acid cycle. I think I gave you a whole table on that. But I didn't talk very much about respiratory quotient, so you don't have to worry about respiratory quotient. I think I did talk about it, but that's okay. Don't worry about that. Reducing equivalence. Yeah, I talked about reducing equivalent. That's going to be an electron, okay? And then coupling reactions, how important coupling reactions are. So in reality, there are no endergonic reactions occurring in the body. Instead, you have endergonic reactions that are made exergonic by a number of different ways. One of them is coupling them to ATP hydrolysis, okay? So we make everything exergonic at the very end, okay? Another way of making things exergonic is to remove the products as quickly as possible. Okay? That's another way of making reactions exergonic. Then I went into detail about glycolysis, the first 5 reactions, the second 5 reactions. I think I highlighted a few of those, okay, so that if I give you the substrates, you should be able to work out the mechanisms for those, okay? Do you have to memorize all those enzymes? No. Would I memorize phosphofructokinase? Yes, okay, okay. Because that one is a biggie, right? That's regulated quite a bit, okay? Then we had a midterm, which seems like eons ago. Yes, that was the first midterm. I had to go away for a bit, so I gave you a video lecture on the metabolic fates of pyruvate, which are, we can list them right here. Those are acetyl-CoA. Wait, what else does it make? Oh yeah, ethanol, right? What does it make? Lactate, very good. So those are the 3 that I can think of. Okay, very good, very good. Oh, and I guess also in theory it can make oxaloacetate as well. Okay, we didn't talk about that, but it can't. Well, we did talk about that in terms of gluconeogenesis, right? So it can make oxaloacetate by being carboxylated. Okay, we talked about methanol metabolism. We did talk about gluconeogenesis. Actually, we talked about all the steps of gluconeogenesis, so not just the first step. Because those are the video lectures. I went into gluconeogenesis, and I explained how for many of the enzymes, you use the same enzymes as you do for glycolysis, but there are a few bypasses that are different. And never do we have gluconeogenesis and glycolysis running at the same time. That would be extremely wasteful. Okay? So we never have those running at the same time. There are ways the body shuts down one while allowing the other one to go forward, or vice versa. I didn't talk about the Pasteur effect. So you don't have to worry about the Pasteur effect. Citric acid cycle, we went over that quite a bit. I even put up a video for you on the citric acid cycle. We talked about regulation of the citric acid cycle. Again, that was that video. I didn't talk about anaplerotic reactions, so you don't have to worry about anaplerotic reactions. Mitochondria. Oh, we spent a lot of time on mitochondria. Whew. Okay. Oxidative phosphorylation, electron transport chain. We talked about different inhibitors of the electron transport chain and oxidative phosphorylation. We talked about standard potential. So those are those E values and those half reactions where you have electrons. And we talked about how to interpret the standard reduction potentials. The more negative, the more ability the molecule is going to give off electrons. Okay? The more positive, it's going to be more accepting of electrons. O2 is very positive. We went over ATP synthase. We went over uncouplers. We talked about that. We did not talk about the mitochondrial genome. So take that off, mitochondrial genome. We didn't get a chance to talk about that. I did talk about heat generation and how those diet pills can cause heat generation through uncoupling. We went through photosynthesis. We went through the light reactions. We talked a lot about photosystem 1 and photosystem 2. We talked about cyclic electron flow. We talked about the dark reactions, the Calvin cycle. Basically, that's just where you're starting to incorporate CO2 and make that into sugar. Okay? We did not talk about the regulation of photosynthesis, so you don't have to worry about that. We did talk a bit about photorespiration, but we did not talk about the C4 cycle, so you don't have to worry about that. Then we talked about lipid metabolism, and we went over all of this, the obesity problem, fat transport, bile salts, lipoproteins. In a video, I talked about cholesterol transport, fatty acid oxidation, fatty acid transport from the cytoplasm into the mitochondria, how that requires carnitine. We talked about beta oxidation quite a bit, which occurs in the matrix of the mitochondria. And we talked about energy output. I spent a whole lecture on the energy output of glucose versus fatty acids. And then more recently, we talked about ketogenesis. I did not talk about fatty acid synthesis. Don't worry about fatty acid synthesis. Unfortunately, I didn't get to that. I didn't get a chance to talk about hormonal control of obesity. It's a very fascinating topic. I didn't talk about control of fatty acid synthesis, so you don't have to worry about that. I did talk about the role of major organs on the control of fuel metabolism. There was this great picture or image from your book, which I talked about. I talked about insulin and glucagon. I didn't get a chance to talk about epinephrine. And I did talk about starvation and diabetes. I didn't really talk about metabolic stress. We talked a lot about biochemistry, and hoping that you guys had a nice kind of whirlwind tour of metabolism. I wish that I could talk in the next course with you. I don't think I'm going to be available, unfortunately, because I've got other teaching commitments that I have to do. I have to teach Introduction to Biochemistry and perhaps a bioinformatics course as well. So actually, those are the two courses that I'm signed up for. So I apologize that I will not be able to stay with you for another quarter. I hope that I've been able to impart some of my love for the subject in this field. And it's a growing field. You guys that want to pursue a number of different professions within biochemistry, keep on studying it, because this is going to be the future. Somebody completely unrelated to biochemistry said, you know what, I've heard that biochemistry is kind of like what computer science was like 20 years ago. I mean, it is just at the cusp of causing major changes in our lifestyle. We'll soon have minute-to-minute monitoring of our health through analysis of our blood and our sweat, and we're gonna know a lot about our own bodies in the future. So with that, we're going to do the instructor review, and I need to give you guys the directions on how to do that. Because as I said, there's been this push to try to get people to do this. So just bear with me so I can get the instructions. Okay. So if you have a smartphone, you can just search for CalCLA to locate the app. Okay. If you have an iPhone, you can just go to this website right here. If you have an Android phone, you can go to this website, but you might just want to search for the app and download it. And then if you don't have your cell phone, but you have your laptop with you, you can also do this by signing in to your MyCSULA identity account and then select the student course evaluations, and then you can just choose this course and evaluate. Your comments in terms of how I can improve the course would be very helpful, especially the technology aspect. This is the first time I'm doing this with running these videos, and I don't know if they're helpful, if they're not helpful, if they're a waste of time, but I would like to have some feedback in terms of how I can improve those. So I'm going to turn that off.